Since new therapeutic compounds are needed for many CNS diseases, where mostly symptomatic treatments are available, different approaches have been used to overcome the limits that blood brain barrier possesses for compounds intended for CNS diseases diagnostic and therapeutic procedures to reach their target sites. One of the most promising alternatives is the use of nanoparticles to deliver such compounds through the blood brain barrier into the brain. Nanoparticles have the same limitations as other xeno compounds to cross the blood brain barrier and accordingly to access the brain. Several strategies have been advised to improve blood brain barrier crossing by nanoparticles, taking advantage of the different physiological mechanisms underlying these processes. The main approaches that have been used to overcome this problem are provoking a transient permeability increase in the blood brain barrier paracellular pathway. The disruption of the tight junctions between adjacent endothelial cells is generally achieved by using ultrasound or microbubbles and osmotic pressure, both of which locally enhance blood brain barrier permeability, allowing an increase in nanoparticle entry. However, this approach entails some risk since it would allow an uncontrolled entry of many compounds into the brain due to the loss of homeostatic, homeostatic function of blood brain barrier, which could lead to cerebral toxicity. Take, Taking advantage of where the surface properties of the nanoparticles facilitate the binding of nanoparticles to the surface of endothelial cells, making use of two different mechanisms. One is adsorptive transcytosis, where the surface properties of the nanoparticles facilitate the binding of the nanoparticle and its cargo to the luminal plasma membrane of endothelial cells. Positively charged nanoparticles are more prone to undergo this mechanism when compared with neutral or negatively charged ones. Since endothelial cell plasma membrane is negatively charged. In addition, nanoparticles decorated with compounds such as wheat germ or glutenine can be taken by nerve terminals and retrogradely transported by the axon to the CNS. Second is receptor-mediated transcytosis, where nanoparticles decorated with different ligands on their surface are able to bind specific receptors and thus promote endocytosis. Several receptors and transporters have been used as targets such as GLUT1, lactoferrin, transferrin or different peptides including angiopep2 or SEC12. The initial mechanism involved in transcytosis is nanoparticle uptake by endocytosis, which consists in the production of endocytic vesicles generated from plasma membrane invaginations, which in turn allow the incorporation of extracellular substances into the cells. It is a complex process involving many components such as receptors and different signaling pathways. There are two main routes of endocytosis. One is Clathrin-mediated endocytosis and second one is Caviolin-mediated endocytosis. First one is Clathrin-mediated endocytosis, which takes place in Clathrin-enriched areas of the cell membrane, forming endocytic vesicles up to 200 nanometer in diameter. Once inside the cell, Clathrin-coated vesicles fuse each other, generating an early endosome that progresses to late endosome while its intravesicular pH decreases and finally fuse with lysosomes, causing degradation of their cargo. In order to be a successful delivering agent, the nanoparticle should facilitate its cargo escape from the endosomes before they fuse with lysosomes, thus avoiding the possible cargo degradation. Second one is caveolin-mediated endocytosis, which takes place in lipid rafts where plasma membrane invaginations of about 80 nanometer in size are produced. After this initial step, caveolin vesicles fuse with other caveolin vesicles, generating caviosomes which avoid the lysosomes and whose final fate will vary depending on the cell type. The interaction of the different nanoparticle types with these two endocytic mechanisms plays a relevant role on the final ability of a given nanoparticle to successfully cross the blood brain barrier.